All right, so welcome to the October monthly meeting of EPAASM. Today we have Dr. Georgina Purdy from Oregon Health and Sciences University. So Dr. Purdy is the ASM Distinguished Lecturer. So let me talk briefly about the program. So the ASM DL program has been around for more than 55 years, and it's provided more than 600 lectures. And it's provided by the ASM and for the ASM branches. And it's funded by the ASM as well as the Whitman Foundation for Microbiology. So that's a very smooth transition of the importance and supporting ASM, as well as our local EPAASM branch. So ASM provides you with your community, your career, your science, providing and nurturing the network as well as your community. We have a new platform for kind of online connection, the ASM Connect. You can join the conversation, you can network between peers, you can share your science, and they're member-only events. And I think ASM is still conducting this salary survey, so if you're interested, you can go on there, asm.org/survey. And I want to highlight two upcoming events for EPAASM. So this is the last monthly meeting for the year, for 2020. In next Friday, we have our 50th annual clinical symposium, our 50-year historic look back and the view ahead for novel diagnostics. It will be a virtual meeting, and so we have a very impressive list of speakers. If you're interested, please go on our website, epaasm.org, to find details and register for the meeting. If you know someone you might be interested, please spread the word. Next, in December 14th, which is a Tuesday, we have our 30th annual Philadelphia Infection and Immunity Forum. Again, it will be featuring invited speakers as well as student speakers and poster sessions. The abstract submission information will be sent out tomorrow. Please participate. As you all know, for the Zoom etiquette, we'll hold all the questions to the end. And you can either use the participant tab and raise a little blue hand for the questions, and we'll unmute you so you can ask your questions. Or you can type your questions in the chat box and we'll relay the question. So I last mentioned that Kyle Rodino and Jason Eindel has joined the program committee. And so Jason will introduce the speaker for today.
Great, thank you. So it is my pleasure to introduce today Dr. Georgina Purdy, who became in 2020 an ASM Distinguished Lecturer, although we just found out that this is her first lecture as DL, at least for this year. So congratulations to us for getting her on Zoom for another Zoom meeting. Very pleased to have her, of course. So presently, she is now, as of 2021, recently promoted to full professor at Oregon Health and Sciences University in Portland. She's in the School of Medicine in the Department of Molecular Microbiology and Immunology. And she joined the department in 2008 after a postdoctoral stint with David Russell at Cornell, which is where she got interested in, presumably where she got interested in and started working on Mycobacterium tuberculosis. Prior to that, she did her doctoral work studying a former favorite pathogen of mine, Shigella flexneri, and looking at how some periplasmic chaperones and proteases contribute to polarization of a protein called ICSA and intercellular spread of that organism, which is a great model system. And then before that, really interestingly, she did her undergraduate degree in Gainesville, Florida, where she got both a Bachelor of Arts in History of Science and a Bachelor of Science in Microbiology down at the University of Florida. And so she really has published a number of excellent papers recently, has dozens of manuscripts to her name, a solid funding record. And what's also interesting to me in particular is that plays a big role in education and the graduate programs at OHSU, in particular as the director of the Molecular Microbiology and Immunology graduate program and as the graduate program in Biomedical Sciences. So I think she'll give us quite an interesting talk combining some biological chemistry, cell biology, and straight up bacteriology on Mycobacterium tuberculosis, the great name, the big ball of wax. So Dr. Purdy, thank you for coming, and you can take it away.
Great, thank you for that really wonderful introduction. I am going to share my screen and hit play, and everything's going to go swimmingly. All right. So thank you again for inviting me to present at your ASM branch meeting. I've been an ASM member since I was a wee young grad student, and I think that the society has a really valuable role to play in our lives as scientists and as academicians. So let's get started with some science. So today I'd like to tell you about a long-running project that's been going in my lab, and it's focused on this family of membrane transporters, Mnpl proteins, and their role in cell envelope biogenesis, virulence, and non-replicating persistence. So Mycobacterium tuberculosis is the causative agent of tuberculosis, and it's a global health concern. Roughly 1/3 of the world's population is infected. We have a number of new cases reported annually. This is likely going to get worse with COVID and interruptions with healthcare. But a really important aspect of tuberculosis is that most people actually have latent TB infection. So what do I mean by that? Well, let's think about TB disease terminology and a little bit of epidemiology for a second. So in terms of TB infection, we know from household contact studies where you're basically tracking family members and household members in the same space as an infected individual, roughly 50% of those household contacts get infected. And that's actually pretty impressive. And that you can really chalk that up to an effective innate immune response. Of those individuals that get infected, about 95% of those develop latent disease. And this is really as a result of an effective adaptive immune response. And so in that latent infection, the bacterium is effectively walled off in a granuloma, and it can you can have a granuloma and still be latently infective and not capable of spreading disease for the rest of your life. And that happens about 95% of the time. About 5% of the time you get progression or reactivation of disease. And so in latency, the infection is controlled, but that bug is still there. It just happens to be persisting in a quiescent, or what we typically refer to as a non-replicating persistent form. And so it's really important to understand that persistence in terms of infection and control of TB. Much of what I'm going to talk to you about today really focuses on the mycobacterial cell envelope. So I'm sort of giving you guys the cartoon version over here on the right. The TB cell envelope is, let's see if I can, yes, I can. The TB cell envelope is very unique in architecture and composition. And so just to walk you from the cytoplasm out, we have a plasma membrane. We have a layer of peptidoglycan. That peptidoglycan is covalently attached to a molecule called arabinogalactan mycolate that forms the inner leaflet of the mycomembrane. And then we have an outer leaflet that is non-covalently associated trehalose dimycolate, or cord factor. And we also have some species-specific free lipids that are either embedded or associated with what some people refer to as a capsule out here on the outer edge. And so there's a number of unique molecules. For instance, this trehalose dimycolate is very characteristic of TB. The cell envelope is crucial for intrinsic resistance of the bacterium to antibiotics and to the host. And some of these lipids are very immunomodulatory. In particular, TDM is very inflammatory, and other lipids we know are involved in host pathogen interactions, for instance, Pbs. And individuals have been studying the mycobacterial cell envelope for years, and I can tell you that there's still unanswered questions about the composition. I feel like we identify a new cell envelope component every couple years, and how the bug builds its cell envelope is still something that we're learning about. Oops, there. So this family of proteins, the Mnpl transporters, plays a key role in the biogenesis of the bacterial cell envelope. So Mnpl proteins are members of the R&D family of exporters. These are large proteins, 12 transmembrane domains, and typically they have a couple of either cytoplasmic or paraplasmic loops. And we've learned a lot, I would say, in the past 15 years about the Mnpl proteins and their substrates. And so I'm giving you an idea of just the substrates that have been identified. The Mnpl proteins sit here in the plasma membrane and are able to export specific substrates to the mycobacterial envelope. And so Mnpl3, which we'll talk about a little bit today, exports trehalose monomycolate. Other hydrophobic substrates, such as Mycobacterium, siderophores, are also exported by Mnpl proteins. These are some of these species-specific lipids here. And Mnpl3 is essential, which is great because it might be a really good target for novel therapeutics. And we know that Mnpl4, 5, 7, 8, and 11 are required for virulence. So this is a really important family of proteins for the bacterium. My lab has spent most of our efforts focusing on Mnpl3 and Mnpl11. These are conserved across mycobacteria, including Mycobacterium leprae. They're always in the same genetic locus. There's a couple extra genes here in Mycobacterium smegmatis, but they're throughout the genus, and they contribute to replication and persistence respectively. So I mentioned a couple slides ago that Mnpl3 is essential and it exports trehalose monomycolate. So trehalose monomycolate is then used as a substrate by the antigen 85 complex to generate that trehalose dimycolate TDM molecule and the arabinogalactan mycolates that form the inner leaflet. So that it totally makes sense that it is essential. And we've learned a lot about Mnpl3 using conditional expression systems. And so my lab and other labs have used this approach to identify the substrates of Mnpl3 and look at the function of this protein. And so in our system, we can conditionally turn off Mnpl3 in the presence of anhydrotetracycline, ATC. And so normally you get TDM, but if you block Mnpl3, you don't get any TDM because you've not exported your trehalose monomycolate. And we can look at bacterial viability as another readout for Mnpl3 function. And so in this case, we're streaking bacteria to a plate, and in the presence of ATC, you don't get growth. We can actually add back an episomal construct that carries the Mnpl3 gene from TB. This is a SMEG system, but we can heterologously complement that and restore growth using that plasmid-based system. So that was Mnpl3, and it contributes to replication. What I'm going to spend a little bit more about time on today is telling you about Mnpl11 and how it contributes to persistence. So non-replicating persistence, as I alluded to before, is really important because most people with TB have latent infections. those persistent bugs that are within the host have distinct cell envelope composition and are really important, phenotypically resistant to first-line TB drugs. So this definitely contributes to the length of TB therapy, which is 6 to 9 months, multiple antibiotics. So the really big questions in the field and the things that we think about in my lab is, how does TB establish non-replicating persistence? What pathways are still functional? Because these aren't dead bugs, they're still metabolically active. What pathways are functioning to keep them that way? And if we discovered these pathways, can we target them with new therapeutics? So my lab uses 2 in vitro models for non-replicating persistence, and they both incorporate nutrient and oxygen restriction, which are 2 sort of triggers for the establishment of dormant bugs. They just do it in different time frames. So the first model that I'm showing you is a biofoam. And so I've got my Corning bottle over here. This is a 4-week TB biofoam. The bugs sit there for a while, and then in about 3 weeks, a pellicle, a layer, forms at the air-liquid interface. I sort of refer to this as that layer that forms in your coffee cup when you've got cream in it and it gets cold, and then you reheat it in the microwave, and then you've got this like nasty plug of stuff that, if you're me, you reach in and you grab it and you fling it. So similarly, we reach in and we grab that pellicle, and we can look at what is in that biofoam. And so in doing so, we know that it's got a lot of lipids. Extracellular material comprising that pellicle includes free mycolic acids, long chain tags, mycolate wax esters. We can also look and see what the bug is thinking and doing, because some of these bacteria are really persistent and some of them are just somewhat less active. Our second model for non-replicating persistence is what we call Pbs starvation. So we take growing bugs, and our normal media is 7H9 medium, and we wash them in PBS, and then we resuspend them in PBS and put them in a tube with very little oxygen. So all of a sudden, very rapidly, they experience nutrient restriction and oxygen restriction. And so both of these models are really good at generating phenotypically resistant bacteria. So we've got a couple of first-line TB drugs here, RIF, INH, ethambutol. And if you have 7H9 replicating bugs, you can kill them upon treatment with RIF, INH, or ethambutol. If they're sitting in PBS, you cannot kill them, because nothing drastic has happened. They don't have mutations in RPOB, but they are phenotypically resistant. And so really what we need more of probably in the field are compounds more like this one, which we characterized and showed that it actually was effective against dormant persistent bugs. Okay, so I'm going to tell you 2 main stories today. And the first story is about Mnpl 11 and what we learned about it in terms of its substrate and its role in non-replicating persistence, and some thoughts that we have about what those Mnpl 11 substrates are doing. So let's jump in here. And please, guys, if anyone had, I know that Vincent said to not interrupt, but I sometimes feel like these Zoom seminars can go very long, and I'm happy to be interrupted at any time. Okay, here we go. So one of our first observations about Mnpl 11 was in Mycobacterium smegmatis, and we saw that the Mnpl 11 mutant has reduced biofilm formation. So these are 4-day-old biofilms in Petri plates, and I've zoomed in a little bit so that you can see that in the wild type bugs, we have a pellicle and it's reticulated. It forms really nice structure. The Mnpl 11 mutant has a very weak biofilm and it doesn't reticulate very much. And we can complement that. This happens to be an episomal vector expressing Mnpl 11. I had a technician at the time, Sophia Pacheco, who did some beautiful transmission EM of these biofoams, and you can really appreciate the cording and the structure of these biofoams. The 11 mutant has very flat structure. It doesn't have pockets of gooiness like this guy does. And you can really even appreciate that those bugs seem to be a little bit more naked compared to the wild type and the complemented one. So that was our first phenotype for this guy. And we really went through a big process of lipid analysis. And I'm showing you thin layer chromatography, TLC plates here, where we've extracted total lipids as well as just the surface exposed lipids. And you can see that we have a couple species that are absent in the Mnpl 11 mutant that are present in the wild type and complemented strain. Using mass spec, we identified these lipids as a meromycanyl diacylglycerol, which is basically a tag or a triacylglycerol, except for it has a meromycolic acid on one of its R groups. And the other lipid is this mycolic wax ester, which has a wax esterified to a big mycolic acid. So these are really just big molecules. And so you can imagine just like thinking about these big waxy things, how the heck do they get anywhere? Well, apparently these two need Mnpl 11. So based on this data, we developed this model where Mnpl 11 transports mycolic acid containing lipids in Mycobacterium smegmatis. So in the cytoplasm, you synthesize your mycolic acids. At a certain point, you either go towards trehalose monomycolate, which is going to eventually become trehalose dimycolate, or you become MMDAG or mycolate wax ester, and you go out through Mnpl 11. Both of these transporters that we're interested in, very similar to one another, co-located in the genome, have similar substrates, but can't complement or compensate for one another. Okay, so that's in SMEG. And SMEG is a really great non-pathogenic model where sometimes you can bootstrap your way into the answer to your question. But our main question is not about what is Mnpl 11 doing in SMEG. What we really want to know is what is it doing in TB, right? And so just starting out, we were able to express the Mnpl 11 gene from TB in our Mnpl 11 SMEG mutant. And so based on that heterologous complementation, we hypothesized that Mnpl 11 transports mycolic acid containing lipids in TB as well. And we expected that the 11 mutant would have altered phenotypes, including biofoam formation. And that's actually, it's great when science works like it's supposed to. So these are TB biofoams. I've grown them in 6-well plates instead of the Corning bottles so that you can get eyeballs on these qualitative observations that this is our wild type biofoam. It's beautiful, it's nicely reticulated. This is really weak, like you can see through it. And then we've got complementation both by a plasmid vector as well as an integrative construct. Biofilms, typically, we also can look at biofoam formation and the strength of the biofoam using crystal violet staining, which just is sort of non-specific, but it works. And you can see that you have less crystal violet staining of the Mnpl 11 mutant, and it's complemented. And it's really, this is really important down here. We actually harvested these biofoams and mechanically disrupted them and then plated serial dilutions. And it's really important to note that there's the same number of bugs in here as there are in here. They're just floating around planktonically and not present in that pellicle. We looked at lipids again. So we're looking at our TLC plate here. We've got our surface exposed lipids and our total lipids, and you can see 2 species that are missing. And we identified those as a mycolate wax ester, slightly different flavor of wax, but that was there. And then a long chain tag. And so in SMEG, we had a mycolic acid here. In TB, it's a fatty acid that has 20 to 38 carbons. Turns out that is actually a big species difference. We've never been able to see MMDAG in TB, and we don't see this long chain tag in SMEG. So reasons to work in both systems. Okay, so based on those data, we modified our model, and we have mycolic acids / tags in TB that are being generated, and the long chain tags and mycolate wax esters are going out through Mnpl 11 to get incorporated into the cell envelope. So that's all well and good, but why is Mnpl 11 required for virulence? Previous work from a couple of labs had shown that there was an attenuation of an Mnpl 11 mutant. One study showed that while the Mnpl 11 mutant infected and replicated initially in the mouse, it failed to persist, and bacterial numbers dropped after 21 days, which is about when the adaptive immune system kicks in, and just those mice had a much longer time to death than those that were infected with wild type TB. And so consistent with that, when we infected THP1 monocyte-derived macrophages with the Mnpl 11 mutant, we don't see any phenotype. There's really no defect. However, we used an in vitro granuloma model where we infected PBMCs from latent TB individuals. And so you're going to have monocytes in there, but you're also going to have TB reactive T cells. In that context, we do see that the Mnpl 11 mutant is more has reduced survival compared to wild type, and that is complemented. Another aspect of the bug that might contribute to its virulence is its lipid content. And so in vitro studies had shown that tags and wax esters are synthesized in hypoxic conditions, so that's sort of like a dormancy trigger, and that they are required for TB to enter in vitro dormancy. And so we thought that maybe the Mnpl 11 mutant might lack these lipids, these extracellular storage lipids that are required to establish or survive in persistence, and that might affect the virulence of the bug. And so to look at that, we used our Pbs Starve model, and what I'm showing you is wild type mutant and complement over 6 weeks in that Pbs starvation. And you can see at 6 weeks, these bugs have very much reduced survival. The colonies were actually super tiny, and we took this out till 9 weeks, and we were able to recover wild type bugs, but we weren't able to recover any of the Mnpl 11 mutants. So it definitely has reduced survival or ability to resuscitate from this in vitro dormancy model. So how do these long chain tags and mycolate wax esters actually contribute to TB non-replicating persistence? And I think that's a really great question, and our thoughts about this are sort of informed by some recent studies in the field that have looked at lipid composition and lipid recycling in terms of TB physiology. And so this cartoon is summarizing some data from both clinical samples as well as metabolomic studies that have been performed in TB. And what we know from infected tissue or sputum is that dormant bugs have decreased acid fastness. Acid fastness is correlated with mycolic acid, TDM content. and reduce cell wall permeability. And what is thought is that the trehalose monomycolate and trehalose dimycolate are actually hydrolyzed and release free mycolates and trehalose. The trehalose is imported back through this LPQY sugar ABC transporter. And metabolomics studies actually show that that trehalose gets pulled into pentose phosphates during dormancy. In addition, there's these little white blobs in my bug, and those are actually intracellular lipid inclusions. These lipid bodies actually contain tags as well as some other lipid components. And so this is what our dormant bug looks like. But when we resuscitate, so basically the metabolomic studies took the hypoxic bugs and then re-aerated them, what we find is those pentose phosphates get shunted straight into peptidoglycan biosynthesis and contribute to building of new cell wall and replication. And then you also have to remake your TMM and your TDM. And when this story came out, myself and I know other people were like, oh my gosh, you just spent so much energy making these big waxy or complicated molecules. You're going to send them out and then you're going to degrade them. Oh my gosh. But this happens apparently. Okay, so how did that impact what we were thinking? Well, we also have been thinking about how lipids change and are they recycled. So in the lab, we were looking at biofilm formation, but then also dispersion. And so we grew up our biofilms in transwells and they look cute and beautiful. And then we took the transwell and we dip and transferred it to new growth media. And so we've got our planktonic profile here. This is our biofilm profile. So you can see that they are very distinct. And within 4 days of dispersion, at which point the bacteria maybe have gotten their acts together and replicated once, those lipids are gone. Okay, so what happens to them? Where did they go? One thought that we had was that we really didn't know what was happening in the bug at this point in time. So in order to get an idea about the genes involved and the pathways involved in lipid metabolism in dormancy versus planktonic growth, we performed RNA-seq and we identified 339 genes that were differentially expressed in TB biofilms compared to planktonic culture. And you won't be surprised that there's a lot of differences in metabolism and cell wall processes. But what did we learn from this? So there were some really interesting genes that were very highly upregulated in our biofilm RNA-seq. And I'm showing you the qRT-PCR data where we confirmed their upregulation in biofilms, and we also looked to see if it was similar in our Pbs starvation model. So one of our top upregulated genes in biofilms is this RB2301, which is a secreted hydrolase. We also saw high upregulation of a triacylglycerol synthase, a tag synthase, TGS2. And we saw tenfold upregulation of an MCE1 locus that encodes for a fatty acid import system. We looked at a couple of genes that we knew didn't move just to have a negative control here. But based on this, we have a working model that we're currently testing that to survive within biofilms or during non-replicating persistence, TB reprograms, it upregulates the biosynthesis of long chain tags and mycolate wax esters that it then exports through Mnpl 11. These mycolate wax esters and long chain tags are then hydrolyzed, perhaps by cut2. I think it actually, I can tell you that it's not just cut2, there's others involved too, into the wax, the free mycolic acid and fatty acids, which are then imported back into the cell and could be incorporated directly into new cell envelope lipids, or it could be beta oxidized for energy and nutrients. So we decided to take a genetic approach, and we have constructed mutants in these genes. And I'll show you one bit of data that I find validating. So we've got wild type, we've got an MCE1 mutant, we have the complemented strain. We're looking at surface lipids and total lipids and biofilms. And you can see that we've got a huge accumulation of something. And it turns out that those are free fatty acids that have accumulated in the biofilm. So the bacteria is able to form a biofilm, but it's not able to re-import those fatty acids that are generated in that biofilm. In addition, we find that using the Pbs starvation model, we actually see that the MCE1 mutant, similar to the Mnpl11 mutant, has a reduced ability to survive or resuscitate from in vitro dormancy. So we actually think that these exported lipids are recycled by the bug upon resuscitation. So we've made a number of direct mutants, and those mutants have allowed us to test this hypothesis. I will add that there's about 10 tag synthetases that contribute to tag biosynthesis in TB, and so knocking out one of them is not sufficient. A number of them contribute. There's also a number of secreted hydrolases, and so knocking out cut2 is not in itself sufficient to decrease the generation of free fatty acids and mycolic acids from these mycolate wax esters and long chain tags. We're also using metabolomics to actually track these lipids and see where they go and see if it's being incorporated into new cell envelope lipids or into the beta oxidation. Okay. Okay, so the second story that I'm going to tell you today, and some of this work has been done in collaboration with a structural biologist, Dr. Edward Yu, who's now at Case Western. We've been looking at the structure and function of Mnpl proteins. So I mentioned that Mnpl proteins are R&D family members, but we have some data to indicate that they really are unique in their function. They largely export these hydrophobic substrates, not necessarily antibiotics, which is what typically gram-negative R&D efflux pumps are exporting. Some of them have identifiable, but most of them don't have readily identifiable paraplasmic membrane fusion proteins that form the channel across the paraplasm so that things get effluxed. And intriguingly, the Mycobacterium smegmatis Mnpl3 structure that Ed solved revealed that it was a monomer. And there was another group at the time that also published a structure, and that one is also a monomer. So there's some good agreement on that. So that really makes it distinct. So in terms of this part of my talk, I'm going to tell you about the identification of potential paraplasmic fusion proteins that interact with Mnpl proteins, and tell you about the identification of residues that are important for Mnpl11 function. So this is my cartoon version of our Mnpl proteins. And so we've got a couple of paraplasmic domains and a C-terminal cytoplasmic domain. And to identify binding partners of Mnpl3 and Mnpl11, we used a two-hybrid approach. The MPFC system is a Mycobacterium base system where you've got two parts of DHFR, and you clone your library and you clone your bait. And if you have proteins that interact, you get growth on trimethoprim. And you can measure the strength of the interaction through trimethoprim MICs. Using this approach, we identified in our screen LPQN, a lipoprotein, as a protein that interacted with a paraplasmic domain of Mnpl 11. And so I'm showing you here the Mnpl 11 D2 loop interacts with LPQN. We also checked to see if it interacted with Mnpl 3 D2 loop, because Mnpl 3 and 11 are fairly similar. And we also found that interaction as it conferred trim resistance. So that was really interesting. And then we were looking to see what interacted with LPQN, and interestingly, we found that LPQN interacts with antigen 85. And so TMM, if you guys remember, is exported by Mnpl3, and that antigen 85 complex converts two TMMs to one TDM. And so we think that LPQN is there to facilitate either chaperoning or forms a scaffold to make sure that the substrate gets to where it needs to get. In support of that, we collaborated with Ed Yu, and he was able to solve the crystal structures of apo-LPQN, but also LPQN in complex with two trehalose compounds that are supposed to mimic trehalose monomycolate. And so that really establishes LPQN and perhaps other lipoproteins that are similar to LPQN as a potential lipid binding partner of the Mnpl proteins. So next, and this was sort of ongoing at the same time, we were interested in the identification of residues important for Mnpl 11 function. And for this, we went back to our heterologous expression system where we're in Mycobacterium sphegmatis in the Mnpl 11 mutant, but we're expressing the TB protein. And so biofilm and wild type, not in the mutant. We can restore biofilm formation with Mnpl 11, the wild type protein from TB. And then we had a series of mutations. This first one, Y610F, that tyrosine is known in other MMPLs to be involved in the protein's ability to utilize the proton motive force. So it's part of the proton relay, and that protein is dead, which we anticipated. We also did deletions in the D1 and D2 loops and found that the D1 loop is really important for biofilm formation. By I, the D2 and the C terminal deletion mutants formed a biofilm. It wasn't a great biofilm. We followed up with lipid analysis. These are TLC plates. And so what I'm showing you is that our two substrates of Mnpl 11 are absent in the mutant, complemented with wild type, not complemented with the D1 deletion mutant, and or the Y610F. But all of the TDM is getting out just fine because it's using Mnpl 3. We've really only broken one of our Mnpl proteins here. And importantly, it wasn't because we weren't getting protein expressed. This protein hates to be expressed. I will just say it. We can see protein by Western, our wild type protein. There's actually more of the 610 and the delta D1 loop, but those proteins are just not functioning. A study about 10 years ago from Bob Hussen's group at Harvard looked at the global phosphoproteome of TB in a variety of different conditions. And I like to tell my students and my technicians, if someone publishes a data set, oh my gosh, you have to explore that data set. You just get into their Excel spreadsheets and you find interesting stuff. And that's because I can't afford to do all of the experiments that everyone else is doing, right? So in this publication, the Hessen group showed that there was a threonine in the C-terminal domain of Mnpl11 that was phosphorylated during planktonic growth and not in other stress conditions that they looked at. And so we made phosphoablative and phosphomimetic constructs. And interestingly, we found that the phosphomimetic construct also failed to complement the biofoam phenotype or the lipid phenotype of the Mnpl11 mutant. So this is in our SMEG system. We definitely are getting protein made. It's just not functioning. And so we went into TB with this construct. We've got wild type H37RV. We have our Mnpl11 mutant that has a very weak biofoam. It's complemented very nicely by the T765A, but not by the T765E construct. Our lipids look how you would expect. The A restores to wild type, the E mimetic does not. And again, this is not a general defect. Mnpl3 is working just fine. The rest of the lipids in the cell envelope look normal. This is really just a point substitution that impacts the function of Mnpl11. Interestingly, this is our Pbs starvation model, and turns out it's not important, that phosphorylation is not important in this model. And that's actually a little bit interesting to us because slow transition versus a fast transition, maybe the phosphorylation signaling pathways are a little bit different. So what is the role of this phosphorylation and what do we think is happening? So we found that Mnpl11, when it's expressed from its native promoter, accumulates over time. So this is at increasing ODs. We see an increasing amount of the his-tagged Mnpl11 quantitated below. However, when we look at the levels of T765A or E, we find that normalizing it to GROEL, we find that you don't see the gradual increase over time. And in fact, the phosphomimetic appears to be lower in general than the 765. So this is, if you want to look at it as levels for each allele relative to OD of 1, we're just at 1, as opposed to going up threefold. So it looks like the phosphorylation impacts protein stability, which is interesting. We've spent a lot of time looking at Mnpl expression, not just Mnpl11, but all of our Mnpls. And we find that Mnpl11 is not significantly regulated at the transcriptional level. It's not going anywhere. However, data I just showed you, as well as work from others, has shown that the levels of Mnpl11 do change over different conditions. And so this is data from Schubert et al. That did quantitative proteomics, and Mnpl3 is in blue, Mnpl11 is in orange. And you can see over a time course of hypoxia, where you're going from day 0 to day 24 hypoxia, you see an increase in the amount of Mnpl11, and it rapidly drops after resuscitation. And so our hypothesis is that Mnpl levels are actually regulated by phosphorylation. Mnpl11 is phosphorylated in planktonic growth, where it's not needed, and so turnover is high, and it is not phosphorylated in biofoams, where you want the protein to accumulate and export those biofoam-associated lipids. So in our structure function studies of Mnpl proteins, we've shown that Mnpl 11 utilizes the proton motive force. We've identified lipoproteins that appear to interact with Mnpl proteins to facilitate lipid export. We've demonstrated that phosphorylation of Mnpl 11 impacts the function of that protein, and we think it actually impacts protein levels. And so if you don't have that transporter there, you're maybe not synthesizing these, or they're going elsewhere. The mycolic acid is probably going out as trehalose monomycolate. And in future work, I think that this is actually something that we're going to investigate further. We're really interested in identifying which of the kinases phosphorylates this residue. So TB has 11 serine threonine protein kinases that could be doing the job. We have an idea of which one or which ones might be doing the work. And we also think that there's phosphoregulation of other Mnpl proteins. There's data in the literature that hints that this happens with Mnpl 3 and with Mnpl 7. Other groups have identified phosphosites in those proteins. And we're still interested in understanding a little bit more about lipoprotein adapters for Mnpl function. Some tantalizing new data. We used our SMEG conditional strain, and we expressed the Mnpl 3 containing point substitutions at a couple of the phosphosites, and the threonine 841 doesn't seem to be impacted. This is our empty, so you can see how you kill it if you don't have a wild type copy. 840A doesn't seem to impact growth, but 840E does. On the other hand, this is a biofoam, and we've quantified biofoam material using crystal violet. This is how much you get, and we do the knockdown of the Mnpl 3 at day 2, and day 3 is when the pellicle forms or it doesn't form. And so you need a wild type Mnpl 3 to form a good biofoam. And it looks like the 840A doesn't function as well in that context. So there's something going on, and I'm keen to figure it out. But for now, I will wrap up by acknowledging the folks who have done the work. A lot of the lipoprotein and the Mnpl 11 point substitutions, that was the work that Jeff Melly did as part of his PhD dissertation. Current lab members, Merritt and Carey, are busily cloning point substituted constructs and playing in the BL3 with biofoams and lipids. And we have great collaborators, in particular, Dr. Fangfu Xu at WashU is my long-term lipid mass spec guy, and these are not trivial things. It's not as if when you send a proteomics sample and you get mass spec back. There's a lot of voodoo and creativity involved in identifying and characterizing novel lipids. So with that, I will stop and take questions. Let's see.
Thank you for a wonderful talk. Let me ask the first question. So I'm not too familiar with the in vitro granuloma model where you infect PBMC from latent individuals. How do you think the T cell, MTB specific T cells would affect the MMPL importance in the survival of the TB?
So in this case, this is an in vitro granuloma model that Larry Schlesinger actually set up, and that was when he was at Ohio State. So I think what is happening is those T cells are seeing TB antigen presented by the monocytes, and they're starting to secrete interferon gamma. although I have not ever just hit the macrophages with interferon gamma to exclude other antimicrobial aspects of T cell biology. So, we look at it as immune pressure. In a different granuloma model, we take THP1 cells and infect them. And then our collaborator here at OHSU, Dave Lowenson, has T cell clones that will recognize antigen presented by those THP1s. It's a little bit simpler. And in that context, the T cells are making gamma. They're also words, sorry. At any rate, we know that those guys are making gamma and presumably activating the macrophages to control the replication of the 11 mutants versus wild type.
Okay, so Dear asks, just wondering about your thoughts for MTB metabolism in latently infected humans during tens of years. So longer than time-limited lab experiments. Considering our model, wouldn't, for example, trails become limiting after some time or years for import and energy source during latency survival?
Okay, so trehalose is not, I see what you're saying. So later during dormancy, the bugs theoretically don't have trehalose dimycolate or trehalose monomycolate out on the surface because they've re-imported it and they've put it into a pool. So that's actually good for the bug in the sense that then you have removed a molecule that is sensed by the host, typically through like Mnpl, for instance, which is going to recognize TDM. And so I think it's beneficial for the bug to do this. I think, if I understood your question correctly.
And Sunny asks, thanks for a great talk. Does MTB form biofilms in vivo in the lungs of mice or humans? And does it contribute to the in vivo virulence? And is the Mnp11 mutant more sensitive to killing by antimicrobial peptides, RS, or interferon gamma activated macrophages, or GM-CSF activated macrophages?
So the first question is a good one, and that has come up like relatively frequently. Because I think that people think about TB as an intracellular pathogen and where would it biofoam. So it turns out in the granuloma, you do get free bacteria that are in that nasty goo, and they cord. And you can see this not necessarily in mice, because mice don't form really good granulomas, but you can see this in, I believe it's guinea pigs. And this is work that was done at Colorado State by the Bazarova group, where they actually have histology, and you could see these corded bacteria. And cording is pretty much the same as that biofoam, where you're aligning the bugs and forming these rope-like structures that you could sort of see in the TEM. So that is, I think, becoming more and more accepted and less controversial. And so is the Mnpl11 mutant more sensitive to killing by antimicrobial peptides? That's a funny story, because this mutant, the SMEG mutant, was actually identified when I was a postdoc. I did a genetic screen for mutants that were more resistant to ubiquitin-derived peptides, because when I was in David's lab, we showed that ubiquitin-derived peptides were part of the lysosomal killing milieu. And so to understand how these peptides work, we did a genetic screen, and you sort of, I forget, what's the joke? You get what you're asking for, or you don't know what you're asking until you get the results. At any rate, it turns out that the Mnpl11 mutant has a less permeable cell envelope, and so it's actually more resistant. to those peptides. And we don't see a difference in terms of antibiotics or reactive oxygen. And the interferon gamma activated macrophages just on their own did not, it looked just like the resting macrophages in the sense that it was not distinct from wild type.
Let me ask one more question. So what dictates the specificity of the lipid for the Mnpl transporter? And if with enough drug selections, can there be mutations that maybe allow one transporter to cross export some of the other?
Yeah, so let's see. Whoops, I wanted to go the other way. So there's a couple different subgroups of Mnpl transporters. So Mnpl 3 and 11 are very similar to each other, and then the others look more like the Mnpl 7 depicted here, or a distinct type. And so we don't necessarily know is the answer. In terms of Mnpl 3 and 11, the majority of the, I guess, divergence in the amino acid sequence is in the C-terminal domain. And so I think that might have part of it. And in structures that Ed has done, as well as others in the field, he's actually got a structure where the TMM analogs that we use, the T6D, he has that co-crystallized with Mnpl 3, and so you can see where the substrates bind. I think if we were smart, we might be able to see differences in those binding pockets, but for sure these pockets are pretty large in order to accommodate the lipid species that are going through it. So it might take a little bit of modeling, because we don't have a structure of 11, and then playing with maybe some simulations to see where the molecule can fit and move.
Jason?
Yeah, I have a kind of a background question maybe, but I'm wondering how, the way I'll phrase it is, how dormant is dormant? What I'm thinking is, how much transcription is actually happening in these cells when they're in a persistent state of infection? There's clearly some, you see that, but relative to free living planktonic cells, because I think the post-translational modification, phosphorylation playing a role is really interesting. Do you expect or see transcriptional responses on a relevant timescale?
Are you talking about transcription in our in vitro models? So we have done, and I can't even begin to guess what the transcript levels are in planktonic bugs versus PBS starved bugs. I just know that we have to use a lot more material when we take from PBS starved bugs. So there's definitely, but I don't have a ballpark number on that. So they're not sitting around doing nothing. There are certain things that are transcribed, but when we looked at Mnpl 11, for instance, in the Biofoam or in the Pbs Starvation or in a hypoxia model that a lot of other people use, we did not see Mnpl 11 being regulated at any significant level.
No more questions. Thank you very much for giving a wonderful talk, and thank you for accepting our invitation. And thank you everyone else for joining us.
Awesome. Thank you guys for coming and the invite.
Yeah. Thank you. Bye.
Yep, thanks for coming.